Hi, I'm Dr. Gregory Schimizzi from Carolina Arthritis Associates. Indication extrapolation, or the approval of a biosimilar for a disease which it has not been evaluated in clinical trials, is one of the main reasons biosimilars can be licensed under an abbreviated regulatory pathway. In other words, the FDA might approve a biosimilar for all its reference products indications, even though the biosimilar's manufacturer has only conducted comparative clinical studies in a single indication. Per FDA guidance, indication extrapolation requires adequate scientific justification and will be evaluated by the FDA on a case-by-case basis. This is particularly important in therapeutic settings with curative potential. If a manufacturer wants to develop a biosimilar of a biologic drug that is approved for several indications, which indication should the manufacturer study to demonstrate that extrapolating the other indication is scientifically justified? The FDA recommends manufacturers consider the following categories: MOA, PK biodistribution, immunogenicity, differences in expected toxicities, and any other factor that may affect safety or efficacy. Per the FDA guidance, manufacturers are encouraged to identify a condition of use and the sensitive patient population most likely to detect clinically meaningful differences when looking to extrapolate efficacy data. Regarding scientific justification for extrapolating toxicity data, the FDA recommends manufacturers collect data from the patient population most likely to develop such toxicity. When looking to extrapolate efficacy data, the FDA recommends collecting data from the patient population that is most likely to respond to the reference product. Immunogenicity, or the potential of a biologic product to provoke an immune response, is another concept that is important in the world of biosimilar indication extrapolation. It is only possible to extrapolate immunogenicity data from high-risk populations to low-risk populations, and not the reverse. In some cases, healthy volunteers may be the most sensitive population to study when seeking to reveal differences in immune responses, as healthy volunteers are not immunocompromised and therefore will exhibit normal immune responses. However, if extrapolating immunogenicity data from healthy volunteers does not provide adequate scientific justification for indication extrapolation, these data should be extrapolated from the patient population most likely to exhibit immune responses to the molecule in question. For example, patients on immunosuppressants would be less likely to develop immune responses compared with patients who are not immunosuppressed. FDA guidance recommends the sponsor use a study population sensitive to predicting an immune response and predicting differences across the conditions of use of the reference product. Extrapolating immunogenicity, efficacy, and toxicity data only comprise a portion of the evidence the FDA requires biosimilar manufacturers to produce to scientifically justify indication extrapolation. Other data must also address pharmacokinetic and pharmacodynamic measures, mechanism of action, and any other factor that may affect the safety or efficacy of the biosimilar in each extrapolated indication. How will you know in which patient population a biosimilar has been clinically tested? This may not always be evident from a biosimilar's label. Some of it may be available on the FDA's website, but that will vary by the product. As we have seen, indication extrapolation allows biosimilars to reach the market more quickly. However, given the complexities of the science behind indication extrapolation, it may not be appropriate in all cases, depending on the pathophysiologic mechanism of each disease.